Hey everyone, in this video we are going to take a look at part 2 of the central nervous system, which, sorry, let me rephrase that. Part 2 of the central nervous system book, which is chapter 12 in the Human Anatomy and Physiology by Marriott Van Hoen textbook. About 75% of this presentation is in reference to the spinal cord. So I kind of think of part 1 being brain and part 2 being spinal cord. That said, the first several slides actually are in reference to the brain, protection of the brain and some disorders of the brain. So let's go ahead and take a look at that. So we can't really talk about protection of the brain without talking about the meninges. Of course, the skull plays a huge role, but the meninges do as well. You have 3 meningeal layers, 3 layers of these meninges, and we're going to kind of talk about what those are and what the individual characteristics are of each. They all cover and protect the CNS. You'll find blood vessels between them, within them, as well as venous sinuses, specifically in the Dura Mater, and we'll explain what that is as well. They contain the fluid that your central nervous system is suspended in. They also form partitions in the skull to help compartmentalize the brain. You'll find, for example, the falx cerebri divides the hemispheres of the cerebrum. The falx cerebelli divides the hemispheres of the cerebellum, and the tentorium cerebelli divides the brain from the cerebellum. Divides the cerebral hemispheres from the cerebellum. So they form these little partitions, and it just helps anchor the brain, keep it from moving around too much, basically. So if we're going superficial to deep, meaning if we were to insert a needle into the skull or into the vertebral canal, we would puncture them in order: Dura Mater first, Arachnoid Mater second, Pia Mater third. Those are the three types of meninges. So let's look at Dura Mater first. This literally translates to tough mother. If you think about the word durable, it's just something that is tough, basically. So Dura Mater, mater means mother, so tough mother. It is the strongest meninx. Meninx is just a singular form of meninge. It is quite tough. We have in the cadaver lab, we have 1 or 2 preserved Dura Mater. It's from the brain. It's where we'll teach those partitions I just mentioned, and I believe it extends to like the brainstem area. But anyway, it is pretty durable stuff, even years and years from when it was obtained. So it's much weaker than it was in vivo in the body. And it is still incredibly strong stuff. It's made up of 2 layers. So one layer is fibrous connective tissue, and that's what gives it so much of its strength. Then the deeper layer is a meningeal layer. So it's more cellular. It's not purely fibrous connective tissue like the fibrous layer. This is the only layer present around the spinal cord, actually. In the skull, you have both layers. And that heavily fibrous layer called the periosteal layer, sorry, I didn't mention that earlier, is actually fused with the skull. It's a part of the periosteum within the skull. These are fused together in most places except for the dural sinuses. And basically what dural sinuses are is they're like, it's almost like a vein. So, for example, in the median longitudinal fissure, I'm going to jump down a few slides. Right here, this is called the median longitudinal fissure. It's that fissure between the 2 cerebral hemispheres. You can see that black space that they've drawn in here and how there's 2 layers. Well, those 2 layers are the periosteal layer and the meningeal layer of the Dura Mater. So you see this here says Dura Mater double layer. And then it shows that there's 2 layers, and you can see those layers split apart right here and form this cavity. That cavity runs longitudinally down the median longitudinal fissure. And cerebrospinal fluid drains into that sinus and returns to circulation, venous circulation. So it's almost like a blood vessel, even though it's really just a gap in the two layers of the dura mater. So that's a pretty important function. And then we already mentioned the dural septa, falx cerebri, falx cerebelli, tentorium cerebelli. You don't need to worry about those. I won't ask you about them. Just know that the dura mater does also form partitions in the skull. Arachnoid mater, which means spider mother, that's a little scarier than tough mother. It's in the middle. It's deep to the dura mater. It's loose. It's not attached to the brain, and it's not attached to the dura mater. There's a fluid cavity above and below it. It does not dip into the sulci of the brain, and because it is loose and because there's a fluid cavity above and below it, it forms two spaces, fluid-filled spaces. One is a subdural space. You probably heard of a subdural hematoma. That is a hematoma within that space, and a subarachnoid space. So the subarachnoid space is below the arachnoid mater, and the subdural space is above and below the dura. Something unique to the arachnoid mater is the presence of arachnoid villi. I'm going to go down to this slide again. It's probably pretty tough to see, and now the quality's just lost. But you can see these two little, almost kind of like mushrooms, growing into this superior sagittal sinus within the median longitudinal fissure. This is a place for cerebrospinal fluid to leave this area around the brain and return to circulation. So we're going to have a slide in two slides from now that talks about CSF, but basically the CSF is made within the ventricles, and it works its way out and around the brain and returns to venous circulation. You don't want your brain sitting in the same old stagnant CSF. You want to keep it circulating. This is where it returns to circulation through these arachnoid villi. Well, and I guess this is the next slide, so we can just stay here. And then lastly, we have pia mater, which means gentle mother. This is delicate tissue. It's very highly vascularized. So this is where a lot of the blood supply to the gray matter of the cortex and all over the brain comes from. Through astrocytes, of course, because we know that blood doesn't make direct contact with neurons. It supplies the nutrition through astrocytes, which are the blood-brain barrier. This tissue does cling to brain tissue. It's the only one that actually makes direct contact with the brain, and it dips into the sulci. So if you were to hold a brain in lab, you're most likely touching pia mater. In fact, you may even see that some tissue, a thin layer of tissue, is peeling off of certain parts of the brain. That's pia mater, separating from the actual nervous tissue. So let's talk about CSF. So CSF is cerebrospinal fluid. It is a blood filtrate. You have a lot of blood filtrates in your body, and some that come out of your body, like sweat and urine. Synovial fluid within your joints is a blood filtrate. All of these blood filtrates are similar to blood plasma in that they are mostly water-based with some ions. This particular blood filtrate has a lot less protein than blood plasma. You would not want it to have what's called oncotic pressure, which means that it attracts water because you do not want the pressure to be very high within your cranium. So it's low in protein. It has slightly different ion concentrations to support the function of nervous tissue, but generally speaking, it's similar to blood plasma. It is a blood filtrate, so that makes sense. And blood filtrate, if you're not familiar with that term, it's just something that leaks out of capillaries. In this case, it leaks out of a special type of capillary called the choroid plexus, which is a plexus of capillaries that produce cerebrospinal fluid. And there's choroid plexus located within each ventricle. And we can see that on this slide here. This is a cool slide. You don't have to memorize this by any means, but you can see that there are these specialized capillary, I hesitate to call them capillary beds, but this plexus of blood vessels that releases cerebrospinal fluid into the ventricles. So you can see some here in ventricle 1. I'm sorry, that's technically the 3rd ventricle. And then you can see some here down in the 4th ventricle. And they release cerebrospinal fluid. And then you can see it follows a path, a somewhat determined path. So in this case, it comes down the central canal, and there are actual apertures in the 4th ventricle that allow it to move back here to the cerebellum, and then it can move around the posterior aspect of the brain. It can move around the spinal cord, around the anterior aspect of the brain. Eventually, it's going to return to the sinus, which returns to venous circulation through what's called an arachnoid villus, which we've talked about the arachnoid villi already. And so that is how CSF works. It's released by choroid plexus, and then it's circulated. If you remember ependymal cells, it was a type of glial cell that is ciliated, and those cilia move and push this fluid slowly through the brain and around the brain. And again, you don't want stagnant fluid in your brain. I think the analogy we used a couple weeks ago was the idea of a pond without a fountain. It becomes stagnant, and algae takes over, and different things happen. So that's constantly recirculated. So what does CSF do? It would be easy probably to assume, unless I've said it already, that it provides maybe oxygen, nutrition, glucose, for example, to the neurons, but it doesn't. It does not do either of those things. It does two things. It provides an environment for graded and action potentials to form. So it has a particular ion concentration that lends itself towards that purpose. And 2, it reduces the weight of the brain. Neurons do not do well with compression. Tension is okay, but they do not like compression. Some of you guys have probably experienced neural tension. I remember, I have a problem with tangents. It's better in video. In class, it's terrible. But I hope I didn't say this already. Maybe I did. But I went to public school. I don't know what the breakdown is in this class. Oftentimes, a lot of you guys went to private school, but it's usually kind of a mixed group. Anyway, so I went to public school until Loma Linda University, and we had what was called in public school the Presidential Fitness Test. If you went to public school, you either remember that fondly because you were a strong kid, or it sent shivers down your spine, because those were the tests that every movie references, where you have to climb the rope, and get to the top of the rope. You have to do pull-ups, you have to do sit-ups, and then they score you, like give you a percentile, and if you're high up, you get to brag, and if you're low, everybody laughs at you. I did okay at some things, but one thing I was terrible at was the sit and reach. So I was like a tall, lanky kid, and I was super inflexible. So you had to sit down, like a long sitting position, and you had to lean forward, and there was this little machine that your feet were against, and you had to push it, and it slid, and it measured how far forward you could reach, like how flexible your hamstrings were, basically. And I could not do it very well. Like, I don't even think I could move the little thing that tells you. So I think I got like a 1 inch or something. And the PE teacher came over and was like, "Come on, Trevor," because it reflects on his performance, I think, as a PE teacher, his overall scoring. I'm sure funding is like based on that and stuff. I have no idea. So he was like telling me to try harder and stuff. So I was getting tingling in my feet, like bad tingling in my feet and behind my knees. And I was like, "I can't do it. My feet are tingling." And he was like, "Oh, well, that's just the muscle stretching." And we know, of course, from last quarter that there is no muscle. First of all, what is a sit and reach? What is it stretching? It's hip flexion and trunk flexion too, but you're bending forward. So we know that there's no muscle that goes from your hip all the way to your toes, right? So of course we weren't stretching a muscle, we were stretching a nerve. And we were really stretching the heck out of that nerve because not only were we doing hip flexion, but we were also doing trunk flexion. So the Dura Mater and the spinal cord is being stretched as well. So we're curling up the nervous system and you're stretching it, and it responds by tingling. And in my case, in my feet were tingling. Um, but that's, I got up from the tests and my foot was asleep for a second, but then I lived. Now, the brain does not tolerate compression at all. It can deal with a little tension, the spinal cord, and the brain doesn't really ever have to go through too much tension. It's pretty protected, but the spinal cord does. Neither can tolerate compression. So that is, to bring this back to our class, that is why the CSF is here. Because even under its own weight, the neurons would not be able to survive within the brain. Just the weight of the brain would damage it. So it needs to float. And that's the main purpose of CSF. Okay, so we've already talked about this a bit, but I do want to address again the blood-brain barrier. It maintains a stable environment for neurons, for cell bodies. In the brain, which is the only place the blood-brain barrier exists, the cell that does that is the astrocyte. In the peripheral nervous system, we do have satellite cells to protect the relatively few cell bodies that are in the peripheral nervous system. There aren't many, it's mostly axons. But in the brain, we have a ton of cell bodies. So the entire brain, most of the brain, I should say, there are some exceptions, is sheltered from direct contact with blood because there are things in the blood that could be toxic to the brain. And also we need to control the volume and therefore the pressure within the brain. So we don't just want capillaries that can leak blood plasma, for no particular reason because your blood pressure goes up, for example. So we need a blood-brain barrier. So there are three things that any bloodborne substance must pass through before reaching the neuron cell body. Number one is the endothelium of the capillary wall, which in most capillaries would not be much of a barrier because capillaries are inherently leaky. There are different types. Some are more leaky than others, but generally speaking, they're very leaky. In the brain, they are not, because the feet of the astrocytes release a chemical that causes the formation of tight junctions. Tight junctions, if you remember from our very first lecture, it's like a needle and thread. So the endothelial cells that comprise the capillaries are stitched together. So they're not leaky at all. They're pretty airtight or fluid tight, I guess. There's not much leaking going on. There's also a thick basal lamina, which is connective tissue, around each capillary. So even if they were able to leak through a crack between endothelial cells, we now have a connective tissue barrier. And then they still really can't even get into the neuron at that point. They might be able to leak out into this extracellular space, but in order to get into the neuron, they would have to be actively absorbed by the feet of the astrocyte. So the blood-brain barrier is pretty effective. There are, of course, things that pass the blood-brain barrier. Anything you take orally that affects your brain is obviously passes the blood-brain barrier. So there are, of course, things that get through this. But in general, as long as you don't put anything too crazy into your body, this is a mechanism for protecting your brain. It is missing in places. The hypothalamus in particular is a place where it's missing. And if you think about that, you can kind of start to infer why that might be the case. So the hypothalamus's job is to maintain homeostasis. So it needs to know what's going on in your body to do so. It needs to know what the osmolarity is. It needs to know what the pH is. It needs to know what various mineral balances are. It needs to know these things, oxygen concentrations, right? So one mechanism for that potentially is exposure to blood. And that's the case. The blood-brain barrier is missing there. It's underdeveloped in general in newborns and younger, which is why certain things can affect newborns and younger so traumatically, like smoking and drinking and that kind of a thing. So Anyway, I this isn't funny. I shouldn't laugh, but this picture just cracks me up. It's they had to come up with the mechanism of a TBI, and they used a baseball bat. It seems just kind of silly, but anyway, of course, this could happen. So, TBI, you kind of think of TBI as like a 1 in a million type of thing, but actually, it is the leading cause of accidental death in North America. It doesn't come anywhere close to being the leading cause of death, period, but it's the leading cause of accidental death. And that is primarily because of motor vehicle accidents. We spend a lot of time driving, and accidents do happen, although relatively rare. They do happen, and they can be pretty severe, of course. With any TBI, there's typically 2 injuries, a coup and a counter coup. I need someone to tell me if this should actually be pronounced coup. I've heard these words pronounced in school at one point, but I can't remember for the life of me. So I need one of you smart folks working in the hospital to let me know. But I'm assuming it's coup and counter coup, or contra coup. I don't know. Anyway, goodness. Basically, the idea is that when something strikes your head, or when your head strikes something, your skull remains stationary, and your brain makes contact with wherever the injury is. So this could be a dashboard in the case of a motor vehicle accident, or in this case of the picture, it's a baseball bat. And in this case, the skull is actually pushed backwards, and the brain remains relatively stationary. So the trauma site is anterior in this photo. But then there's a rebound effect, and then so the brain will rebound posteriorly, the head will rebound anteriorly, and you get a counter coup, which is an injury on the posterior aspect in this case. Any injury to the brain that leads to some altered level of consciousness is called a concussion. And they are cumulative, as we know, and there's a lot more research into concussion, with what's happening in football, or at least what was happening before. I don't even know when or if football is starting again, but anyway, a lot of resources have been poured into concussion research, and they do appear to be cumulative, and bad concussions multiple times can have permanent effects on brain function. A contusion, on the other hand, is some type of a bruise, basically, some blood contained within the brain. It's not a bleed necessarily, but there is blood accumulating somewhere in the brain. That can be relatively minor. It can also be incredibly serious. That hematoma takes up space, which means increased pressure, and that can damage your brain. And you've probably heard the term subdural hematoma. That just means it's a hematoma in the subdural space. But it could be subarachnoid. It could be in various places. Any of these things can all be equally severe, but another thing associated with a traumatic brain injury is a hemorrhage. So you can have a subdural hemorrhage or a subarachnoid hemorrhage. And this is a bleed. So it's not like a contusion in that it's contained. It's actually a bleed. So this could also be from, an aneurysm or something unrelated to a traumatic brain injury, but in this context, we're talking about a TBI. And again, the problem here is exposure to blood, so the neurons are exposed to blood, and also increased pressure within the cranium. And increased pressure within the cranium is not a good thing. There can be local injury from the increased pressure, and potentially there could be so much pressure that you could actually do damage to larger areas of your brain, and potentially even move the brain inferiorly, which is what often can cause death in a traumatic brain injury, which leads me to the next item here, which is cerebral edema, which is just general swelling of the brain. So really, these last three things can all cause increased cranial pressure, and if cranial pressure is increased to a very large extent, especially if it's increased globally, like with cerebral edema, the entire brain is swelling, um, the brain can be pushed inferiorly, or I guess caudally, if we want to be really specific, and the brainstem can make contact with the foramen magnum, and that can be what actually causes death from a TBI. Or the injury itself can just be so dramatic that the death occurs sooner. But that's kind of this idea of like the golden hour and getting someone to the hospital quickly so that this can be addressed. Then we have cerebrovascular accidents, CVAs or strokes. These are all kind of synonyms. This is the third leading cause of death in North America. However, this is really not a neurological disorder. Of course it is, because those are the symptoms we see, but strokes are really the same thing as a heart attack. The underlying disorder is the same. We have ischemia or infarction from a clot lodging in an artery, and that artery may be narrowed by plaques and cholesterol-rich plaques, I should say, and that leads to ischemia and infarction and damage. It's a cardiovascular disorder. That said, if we want to be clinically correct, it is a neurological disorder. But I'm just letting you know the underlying physiological issue is the same as a heart attack, actually. So basically what happens with most strokes, there are two different types of strokes. The one I'm going to describe first is ischemic, because it's by far the most common. But anyway, so with an ischemic stroke, basically we're going to learn next week, we have these two structures within our heart. I'm sorry, we're going to learn a few weeks from now. We have these structures within our heart called auricles. A-U-R-I-C-L-E-S. And that means ear, like the auricular surface of the sacrum means ear. And it kind of looks like ears. Every heart looks a little bit different, but I've seen some that actually really do look like ears. Basically, it's just this little pocket sticking out of the top of your two atria, which are the receiving chambers of the heart. And they don't really contribute a whole lot to the function of your heart, but if you have atrial fibrillation, potentially what could happen is that blood starts to pool within these auricles. And when blood stops moving, if you've ever cut yourself and seen blood that stops moving, it coagulates, it hardens up, it scabs up, basically. And those can break off. And this isn't the only source of clots, it's just a very common one. So those can break free, and now you have what's called an embolism. An embolism is a traveling clot versus a thrombus, which is a lodged clot, a stationary clot. Anyway, so you have this embolism floating around. It could go to your lungs, and that'd be a pulmonary embolism. It could go to a coronary artery, and that would be a myocardial infarction or a heart attack. Or it can make its way to the circle of Willis in your brain and lodge in a artery within your brain, and that would be a stroke. It's really the same disorder, it can just manifest in very different ways. So that is called an ischemic stroke. And what happens is blood supply is cut off to a part of the brain. Those neurons are derived of oxygen and glucose and everything that they need to function, and damage is done. And ideally, that clot would dislodge on its own. Typically, that's not the case. So, patient needs to be given thrombolytics, anticoagulants, blood thinners, to alleviate the situation. That is different from a hemorrhagic stroke. It's almost the opposite of a hemorrhagic stroke, but the outcome is basically the same. A hemorrhagic stroke is when a blood vessel ruptures within the brain. This is the idea of an aneurysm. And then we get all the similar symptoms, but caused in a different way. We get blood. Our neurons are exposed to blood, which is toxic to neurons, as well as increased local pressure, which is also can damage neurons. So kind of the two outcomes. What's interesting is you better be sure you know what your patient has, because the treatment for one is the quickest way to kill someone with the other, right? So if someone has a hemorrhagic stroke and you give them anticoagulants and blood thinners, you're basically guaranteeing that you're making it worse, basically. So of course you need to make sure you're addressing the correct disorder. And then lastly, we have TIAs. TIAs are the same as ischemic strokes in that we have a clot obstructing blood flow within arteries. The difference is that this clot never actually becomes lodged and it never fully occludes an artery. It's a traveling embolism. TIA stands for transient ischemic attack. This is typically a precursor to a stroke. So if someone is having TIAs, they'll be put on some type of preventative medication regimen to address this. I can't even remember which one, but one of my former professors told me about an incident he had. He was a physical therapist, and I think he taught one of my cardiopulmonary classes. Anyway, he was working with a patient, and the patient came in because he was falling. He had had a few falls, actually. And so he was interviewing the patient and asking him when he fell. His wife was there kind of translating for him, I think. And so the wife was saying, yeah, he falls all the time. And he's like, well, when's the last time he fell? Oh, he fell just now. Like he fell earlier today. He falls every day, falling all the time. And my professor was thinking, oh boy, that's doesn't necessarily sound like a musculoskeletal issue, but let's, take a look. And so they're talking, and he's having a hard time kind of figuring out a musculoskeletal reason for this to be occurring, or even a, he didn't have a recent stroke or anything like that to explain muscle weakness. He didn't have any muscle weakness. He didn't have any pain. He didn't have any range of motion limitations. He was pretty healthy in that sense. But he would start slurring his words a little bit occasionally. And so his wife at that said, oh, let's say his name's, I don't know, Henry. Henry, stop talking funny. Stop with that. You're always doing that. And my professor was asking, what do you mean he's always doing that? Oh, he just starts talking funny, and I don't know why. And so, fortunately, my professor recognized that as, potential stroke symptoms or whatever. So he advised him to go to the emergency room across the way. And yeah, of course, he was having a TIA, and he had been having them for some time. So he was put on medications to prevent that. So pretty common. Both ischemic strokes and TIAs are associated with heart disease and atrial fibrillation. It's kind of the same underlying mechanism. We are not going to go into much detail about these. I would like to. We may. I don't know. We'll see. So Alzheimer's disease. I think you guys are all very familiar with Alzheimer's disease. A lot of interesting research going on with Alzheimer's disease. It is certainly a disease with the disease process. Part of it, though, too, also is that neurons kind of have a set lifespan. So we're going to see deterioration of the brain in old age, no matter what. Alzheimer's is just accelerated and is associated with certain types of plaques forming, and there is a lot of research in Alzheimer's. I have not stayed up to date with it as much as I should have. Some of my research studies that I'm proposing actually have to do with the elderly, so I should be paying attention. But I'm looking more at inflammation and immunity and those types of things. So I won't speak to that a ton, but you can see right here that you see actual loss of brain mass with severe Alzheimer's disease. And there are various stages of Alzheimer's and dementia. And I'm sure all or most of you have known someone with Alzheimer's disease. I've, of course, worked with lots of patients who have had it. It is progressive, so there really is not a ton we can do about it, unfortunately. Just like these other two diseases, Parkinson's disease. I don't know if many of you guys know someone with Parkinson's disease. It is It's tough. It's tough to watch for sure. It I don't know if any of you had an opportunity to watch that video I mentioned a couple weeks ago about the pallidotomy. Definitely check that out if you haven't. Um, but Parkinson's disease, it has cardinal symptoms. These are common symptoms associated with anyone who has Parkinson's disease, and if you have a certain number of those cardinal symptoms, you're diagnosed. It's not like you do a blood test or something like that necessarily. But anyway, shuffling gait, bradykinesia in general, which just means slow movement, and it is progressive. It does tend to get worse. And I've known a few people with this. My grandpa, who's actually still alive, pretty clearly has this. He hasn't been diagnosed with it necessarily. There's not, at his age, there's not really a ton you can do. He's in his 90s. But I mean, there are certainly medications for this. He's just kind of opposed to that. But the primary one being levodopa or carbidopa or Cinemet is the brand name. And basically, these are all medications that affect dopamine in the brain. And they are effective, but they lose their effectiveness. Your body adapts to them. So a mechanism for combating that is as you go on drug holidays and you take time off, and then you regress, and it's pretty rough on patients, and then you get put back on, and the medication's a little bit more effective. I think I mentioned to you guys, oh no, tangent. I think I mentioned to you guys that I went to Puerto Rico with my friend in school for a clinical rotation, and I did not speak Spanish. I learned a ton when I was there. I've lost it all since, but I didn't speak Spanish. He did. He was always playing tricks on me down there. That my instructor would say something to me in Spanish, and he'd be like, "Hey, she wants you to run downstairs and get, whatever. I can't even think of a walker." And I'd run down and come back with a walker, and my teacher would be like, "What are you doing with that? I asked you to get a clipboard." So anyway, man, my friend's kind of a jerk now that I think about it. So anyway, there was a patient, and he had I guess I'll say white sounding name. And so I can't say it, Leipa, right? HIPAA law. So the teacher was like, "Oh, Trevor, it's your chance. This is your time to shine. Go see this patient." So anyway, this patient, he wanted nothing to do with me, and I don't blame him because he had like end stage Parkinson's, and he was in a bad way for sure. He had really really severe resting tremor, so his hands were just tremoring all the time. He could barely move, and it was rough. It was rough to watch. I did have the fortune though, and I just have to share the story with you because I think it's so interesting. I did have the good fortune to meet his wife, who kind of gave me his life story, and I could not believe it. I could not believe this guy's life story, so I just have to share it. This guy, I'm forgetting it over the years, so this is being recorded, so this is going to be the official version. I'm not sure how accurate it is, but he was I want to say 16 when World War II broke out, and he joined. He lived in, I want to say, the Netherlands, Denmark or the Netherlands. I always mix up Dutch and Danish, but it was one of those two. And he fought in the war, and he was ultimately captured and put into a concentration camp for POWs, I believe. Again, the details have faded, but he escaped with a group of guys, and ended up in I can't remember what country he ended up in, but all of his friends died along the way, and he ended up living. This is where it gets crazy. Living in a cave with a monk. He met a monk. I don't know what country this was. And he lived in that cave for, I want to say, like 2 years with this monk in silence, that they didn't communicate. And eventually the war was winding down or over, and he made his way back to Denmark or the Netherlands, and he bought a boat, bought a sailboat. Don't remember how he got the money. There was some story on the way back where he became rich. I'm forgetting the most important parts of the story, but he had a bunch of money, and he started sailing the world. And the wife was telling me all the countries he had been to. He sailed the world for, I want to say, like 15 or 20 years. He just lived on this sailboat, and he somewhere in that time met his wife, and they lived in Spain for a while, and then he started sailing the world again. So by this point he spoke Spanish, Castilian Spanish, she was quick to point out, and they sailed the world for like another 15 or 20 years together. And then they ended up in Puerto Rico, bought a cattle ranch in Puerto Rico, and he had lived there for like the last 20 years with her. And I was just meeting him there. So it was just such a cool story. But anyway, wow, tangent, I'm sorry. He had very severe Parkinson's, and Parkinson's is a rough one. As is Huntington's disease, although Huntington's disease is much more rare than the previous two. Huntington's disease is sort of the opposite issue of Parkinson's disease. There's two pathways within your basal ganglia. One's called the direct pathway, one's called the indirect pathway. Depending on which of those is affected, movement can either be significantly reduced, which is Parkinson's disease, or amplified beyond your control, which is Huntington's disease. Someone with Huntington's disease is making writhing movements, large swinging motions of their limbs. It almost looks voluntary, but it isn't. They can't control it. And, quality of life is pretty bad with Huntington's chorea, and this can actually affect you at a pretty young age. You can get this as a child, which is unfortunate, but it's kind of the opposite of Parkinson's disease. All right, so now we are at the spinal cord, finally. So we're going to go ahead and look at the anatomy and function of the spinal cord, and sort of set the stage for reflexes, which is going to be our next chapter. So just some basic, this is kind of an ugly slide, I'm sorry, but just some basic factoids about the spinal cord. It begins in the medulla oblongata. I'm sorry. It begins in the foramen magnum. It's as soon as the medulla oblongata ends, the spinal cord begins. Histologically, they're similar, very different functions, of course. The spinal cord extends all the way down to the L1, L2 vertebrae level, which is kind of mid-back. If you find your last thoracic vertebrae that has a rib attached and go one or two segments below that, the spinal cord ends. So it actually ends pretty high. Most people imagine that it goes down to their sacrum. It doesn't. It ends pretty high, and then you have what's called the quada equina, which is farther down on this list, which means horsetail, and it's a collection of nerves that kind of look like a horsetail, I guess, that go from the L2 level all the way down to the coccyx. The spinal cord is a major reflex center. Most reflexes occur within the spinal cord. But also it is a pathway for sensory information to ascend and motor information to descend. So it's composed of ascending and descending spinal tracts. Spinal tracts are white matter, and they're just bundles of axons. The spinal cord itself is protected by the vertebral column, bone, meninges, and cerebrospinal fluid. There is a space outside of the Dura Mater in the spinal cord. The dura isn't adhered to the skull like it is in the brain or, to a vertebra. There is a fluid-filled space outside of the Dura Mater called the epidural space, and that is where an epidural would be performed. The meninges themselves extend all the way down to S2, even though the spinal cord ends at L2. The conus medullaris is the end of the spinal cord. It cones towards the end, and the quada equina emerges from that cone. Filum terminale is a ligament that anchors the conus medullaris to the bottom of your vertebral column, and denticulate ligaments hold the spinal cord in place. There are 31 pairs of spinal nerves, we know a few of them, C5, 6, 7, 8, and T1 from the brachial plexus, but there are 31 total. And if you look at the spinal cord, we used to have a beautiful spinal cord dissection. It was years ago. We can't hold on to cadavers forever, of course, so eventually they are cremated and there's a service for them. I don't know if they're cremated, I shouldn't say that, but there is a service for them. And so we can't hold on to some of these dissections forever, but we did have one, and you could clearly see a cervical and lumbar enlargement. And that's because of the brachial and lumbosacral plexi. There's a lot of traffic to the arms and legs, so it makes sense that the cervical and lumbar regions would be bigger because there's a lot more synapses, therefore a lot more cell bodies, and a lot more action potentials need to be transmitted in those regions. So it makes sense that the spinal cord's bigger. We're going to spend the next couple slides looking at cross-sectional anatomy. Cross-sectional anatomy is its whole own field of anatomy. We're just going to look at a few segments of the spinal cord. And we're going to label different regions as being gray matter or white matter, and then we're going to label what each of those do. So let's get to it. So right here, fortunately they put it in gray, you can see this butterfly shape. That is gray matter. So they're symmetrical, so it's mirror image lateral gray masses connected in the middle by something called the gray commissure. Now within that gray matter we have horns. Unfortunately I can draw. Sometimes I can draw, sometimes I can't. Okay, we are drawing. So we have posterior horns. We have anterior horns. And we have a lateral horn. Now each of these corresponds to a different part of your nervous system or a different function of the nervous system. The posterior horns are all involved in sensory information. Basically, neurons enter here. There's a cell body and the ganglion, and it makes its way to the dorsal horn. So you can think of dorsal horn as being sensory in function. The anterior horn or ventral horn, those words are used interchangeably. Ventral and dorsal those are really terms for animal anatomy, but they've carried over and they're used somewhat interchangeably. Anterior and posterior is technically the correct term, but so anyway, the anterior horn has motor neurons. What we'll learn soon are called lower motor neurons, and they go out to a muscle like that. So there might be, here's your biceps. Look at that anatomically accurate hand. That's beautiful. Okay, so that's motor. So the anterior horn is motor in nature. The lateral horn is motor as well, but it's specifically for the autonomic nervous system. But since it's motor, it goes out the front as well. And it goes maybe to your stomach or something, okay? So those are the horns. Dorsal horn is sensory, meaning that sensory pathways pass through and synapse there. The anterior horn is motor, and the lateral horn is motor as well, but it's motor for the autonomic nervous system. So another way we could say that is the anterior horn contains somatic motor neurons, and the lateral horn contains visceral motor neurons. Let's talk about roots then. We've already kind of spoke about them, but we haven't really laid it out precisely. So we have two roots. These are called rootlets right here. You don't need to worry about those. When I say root, I'm specifically saying, okay, this right here is the dorsal root. Okay? And this right here, oops, I meant to change color. It's not a big deal, but it's going, there we go. This right here is the ventral root. This here, this guy, this is actually a spinal nerve. So let's say this was the C5 segment of the spinal cord. This might be the C5 spinal nerve. in other words, the nerve root of the brachial plexus. That's why root is kind of confusing when we call the brachial plexus roots nerve roots, because technically it is a spinal nerve, and it makes it even more confusing that these are called ventral root and dorsal root, or anterior root and posterior root. So try to keep that separate. Those are all technically correct terms, everything I just said. So you can call this spinal nerve or nerve root in lab when we talk about the brachial plexus. These two are always called roots, though. So anterior root, posterior root, or ventral root and dorsal root. So the ventral root carries the axons from the cell bodies that are located in the ventral horn. So the axons leave the ventral horn and they come out and they go somewhere to the body in the spinal nerve. Okay, so ventral root is motor. If I cut the ventral root, I'd be paralyzed in whatever myotome, whatever muscles this root innervates. The dorsal horn carries sensory fibers, sensory axons. Okay, remember that sensory neurons look a little bit different than motor neurons. They're pseudo-unipolar. So all this little bulge in the dorsal root is a collection of the cell bodies from these sensory neurons. So this might be a pain receptor out in the body somewhere. And you just basically learn most of the structures right there. You learn the gray matter structures, or at least most of it. You learned what the roots are and what they contain. And you learned when they join together, they form a spinal nerve. And it's right about this time at the spinal nerve that they emerge from the vertebral column, and you can see them in a cadaver, for example, if it's dissected well. Very rare that you'd be able to see inside here on a cadaver. It would involve cutting through a lot of bone and stuff. So, not that we haven't done it, but we don't have one now for sure. 4 zones. This just addresses something that we've kind of already addressed, but we're going to add some more detail to it now. So there are 4 zones of the gray matter. Somatic motor, we already talked about that. The anterior horn contains cell bodies for somatic motor neurons, neurons that innervate skeletal muscle. The lateral horn contains visceral motor neurons, cell bodies that innervate cardiac muscle or smooth muscle or glands, and then they might send an axon out the ventral root. That's the only path it can take. I shouldn't say might. It will send an axon out the ventral root. But we did not differentiate the dorsal horn into 2 sections. So technically this more posterior part of the dorsal horn is somatosensory or somatic sensory. So pain, temperature, light touch, vibration, deep touch, from skin, muscle, joints, ligaments, cartilage, that kind of thing. Not cartilage, sorry. Right behind that, near the autonomic motor, the visceral motor, is visceral sensory. Okay, so there's actually 4 zones. Visceral sensory, somatosensory, somatomotor, and visceral motor. Kind of tough. That's all outlined in this picture right here. And they do a good job too of showing you the axons leaving anteriorly, entering posteriorly. That's the flow. Motor information leaves from the lateral and ventral horns out the ventral root. Sensory information goes dorsally. It goes through the dorsal root into the dorsal horn. A little different structure because remember, sensory neurons are pseudo-unipolar, so there's what's called a ganglion here. This structure right here is called the dorsal root ganglion, or DRG. So in a quiz, if I said, hey, what's contained within the dorsal root ganglion? You would say sensory neuron cell bodies. Actually, you'd be even more specific. We just haven't gotten there yet, so apologies, we will. So let's talk about white matter. White matter can be ascending, descending, or transverse. Why does white matter have a direction when gray matter didn't? Well, gray matter was just cell bodies and synapses. White matter is axons. So these axons are going somewhere. They have a direction. They're either carrying information up, carrying information down, or crossing from left to right, right to left. and they're a part of spinal tracts. So there are tracts that carry information up and down the spine. Spinal tracts are a part of a multi-neuron pathway. Could be 2 neurons, could be 3, could be more or less, that connect. I shouldn't say less. I don't think there's any 1 neuron pathways from the brain to the body. There may be. Anyway, so these pathways connect the brain to the body. We can make 4 generalizations about them. The first is that they decussate, and there are exceptions to this, but for everything we're going to talk about, they do decussate. That means they cross. This is why injury to the right side of the brain affects the left side of the body, and vice versa. There is relay. Relay means synapses. One neuron must stimulate another for that information to reach the brain or reach the muscle, depending on which direction the action potential is traveling. There is somatotopy. That means that they are mapped. So, for example, these blue structures over here are sensory tracts. They're ascending tracts. Okay? So if I were to cut this out, first of all, let me just be clear that this entire tract does the same thing. It's responsible for carrying proprioceptive stimuli and discriminative touch to the brain. Sorry, my daughter started crying. I'm back. Where was I? Okay, so we were talking about this region right here as an example. It's a part of a tract, a spinal tract, that carries proprioceptive input and discriminative touch, the ability to tell the difference between a quarter and a penny in your pocket, for example, or a house key or whatever. All that information travels to the brain in axons within this tract. So you can picture axons traveling from a lower level of the body, right? This is behind. We can't really see this. And then they might emerge from here and keep going up to the brain, okay? Because we're just taking a slice out of the spinal cord. So we're picturing something 3D, but we're just looking at a little slice of it. So it can be hard to visualize. Anyway, so that information travels up to the brain. What's interesting though, even though this all carries the same type of input, different parts of this area, this is called the dorsal column, by the way, pertains to different areas of the body. So the axons here may come from the foot, while the axons here might come from the hand, and the axons here might come from the arm. You get the idea. So it's mapped to the body. That's true of all tracts. It's even true of the brain, cortex, the gray matter as well. So we are not going to cover that, but that may be something you would learn in a neuroanatomy class. You would know, okay, this region's cervical, this region's thoracic, this region's lumbar, this region's sacral. And that means axons there are coming from spinal nerves in this part of the spine. So cervical spinal nerves, that would mean that this is the arm, the hand, etc., the thorax, the legs, the lower legs and toes. Okay, makes sense? I hope so. It's somatotopy, the idea that the tracts have mapping. The body is mapped onto these tracts. And then lastly, they're symmetrical. Again, there's a dorsal column here, there's a dorsal column there. You have one for the left and right side of your body. Okay, so we're going to look at 2 of these, 2 examples. We're going to look at a sensory example and a motor example. Okay, so here is, I don't know if this is spinothalamic or dorsal column medial lemniscus. I'm not sure. It's one of the 2. That's not important. It's a sensory pathway. That is what's important. It's an ascending pathway to the brain. Now, this right here is crucial to know. Ascending pathways to the brain are comprised of 3 neurons. It takes 3 neurons to deliver something that a sensory receptor has sensed to the brain. So, let's use an example. Let's use this example right here. There's a foot. This is a picture of a foot, and there's a neuron, a sensory receptor there, and it's kind of blurry. I'm sorry. That's a little nail, for example. I'm going to tell you what I want you to know, so don't stress about it being blurry or anything. This neuron right here is called the first order sensory neuron. You can also call it the primary sensory neuron. First order is preferable. So it's the first order sensory neuron. That's the actual sensory receptor. There's something here in the foot that can sense pain or temperature or whatever the sense is. Okay? So the cell body for this is all important stuff to know. The cell body for the first order sensory neuron is located in the dorsal root ganglion. The first order neuron then synapses on the second order neuron, which is located in the dorsal horn. I drew this a little too far in, but you get the idea, okay? It's in the dorsal horn. That synapses in the dorsal horn. That means that the cell body of the second order neuron is also in the dorsal horn. Okay, the second order neuron ascends, may even decussate. Cross is what decussate means. Oops. And it eventually makes its way to the third order neuron. The third order neuron cell body is located in the thalamus. The thalamus then takes that information to the correct part of the brain for processing and interpretation and perception. That's when you become aware of it, you can localize it, you can tell what kind of sense it is. Did you step on something sharp? Is it a burning sensation? What is it? This is true for any sense, any ascending pathway with minimal exceptions. There are some cerebellar exceptions. They've drawn one in here. We're not going to worry about that. So just to recap, first order neuron cell body is in the DRG, dorsal root ganglion. Second order neuron cell body is in the dorsal horn. Third order neuron cell body is in the thalamus. Okay, so sensory pathways are 3 neuron chains. They have decussation, they have relay, they have somatotopy, and they have symmetry. You know where all those neurons are located. Know the path they take. You don't need to get into detail. Like if this were a neuroanatomy class, you say, okay, this is the medial lemniscus and the medulla, and it crosses along the white commissure. We're not doing any of that stuff. I just want you to know where the cell bodies are, and that's it. I want you to know where the cell bodies are and how many neurons are involved. Okay, let's talk about 2 types. We're not going to talk about spinocerebellar pathways. So let's talk about 2 specific examples of a sensory tract: dorsal column medial lemniscal pathway and the spinothalamic pathway. These are 2 different pathways with 2 different unique routes from their receptors to the brain, but they have a lot in common. They're both 3 neuron chains, and the cell bodies are located in the same place. The first order, remember, is in the DRG, second order is in the dorsal horn, third order is in the thalamus. The path they take through the spinal cord is very different. They decussate in different places, their tracks are in different places, but they're both ascending pathways with 3 neurons. The portion that's within the spinal cord is what we call the tract. And if you look back at the picture here, it's the sensory order neuron that's within the spinal cord. The first order neuron went from the foot to the spinal cord. The third order neuron went from the thalamus to the cortex. The second order neuron traveled in the spinal cord. So when we're talking about a tract, we're just talking about the pathway that the second order neuron takes. When we say pathway, we're talking about all 3 neurons. That's a distinction that I won't probably quiz you on, but just so it makes a little bit more sense. So this particular pathway could be anywhere, in the finger, in the foot, anywhere you have touch receptors or proprioceptive receptors. And it enters the spinal cord and it ascends in the dorsal column. That's why it's called dorsal column. And then later on it travels in the medial lemniscus. That's why it's called the dorsal column medial lemniscus. I don't need you to know any of that part. What I want you to know is what type of sensory information this pathway carries. It carries proprioception and discriminative touch. So if I ask you, a patient has a posterior cord injury and the dorsal, I'll explain what that means. You don't need to diagnose them. And that means that the dorsal column medial lemniscal pathway has been damaged on, I won't even say what side, just it's been damaged. What would they not be able to do? And the question might be, feel pain in their foot or identify an object by touch with their eyes closed. And it'd be B, because discriminative touch is that ability to tell fine detail with your fingers or wherever, or your feet. Your fingers are obviously where you have the most discriminative touch, but you have it all over your body. Or I could say the ability to know where your limbs are in space. That's proprioception. You would lose that ability if this pathway was damaged. Compare that to the spinothalamic pathway. It carries the modalities of pain, temperature, and light touch. And the spinothalamic tract, the second-order neurons of the spinothalamic pathway, travel anteriorly. So if I said, hey, you have an anterior cord injury, which means that the spinothalamic pathway has been damaged, which sensation may your patient not be able to feel? Can they feel pain? No. Can they feel temp? No. Can they feel light touch? No. Could they feel deep touch? Yes. Could they feel vibration? Yes. Could they feel joint position? Yes. Discriminative touch? Yes. So you get the idea. So in your notes, I would just write discriminative touch and proprioception for this one, and pain, temp, and light touch for that one, and you should be fine. So those are just two examples of sensory or ascending pathways. We also have a lot of motor pathways. Going back to this picture, blue are some examples of of ascending pathways, and red are some examples of descending pathways. So you can tell there are a lot of these, and there's more than just what's pictured here too. So, but we're just going to talk about one descending pathway. Most descending pathways, though, follow this scheme. There's an upper motor neuron and a lower motor neuron. The upper motor neuron originates in the brain, in the cortex. It comes down and crosses. In the case of, oh, sorry, it hasn't crossed yet. It crosses right here. In the case of our pathway, it will cross in the medulla at the decussation of the pyramids. We haven't gotten to that slide yet, though. The upper motor neuron synapses on the lower motor neuron in the anterior horn, if this is a somatic motor pathway. And then the lower motor neuron goes out the ventral root, out the spinal nerve to the muscle. Okay? That is a descending pathway. So the example we're going to look at is the pyramidal pathways, specifically the corticospinal tract. So in that case, the cell bodies of the upper motor neuron is in the primary motor cortex, or to a lesser extent, the premotor cortex. But I'll say primary motor cortex for a quiz. It goes down the pyramids, the cerebral peduncles. We've kind of mentioned some of this stuff. It crosses at the decussation of the pyramids in the medulla oblongata. And then eventually, if it's going to the arm, it will synapse at a cervical level. If it's going to a leg, it will synapse on the lumbar level, but it eventually synapses on a lower motor neuron. That lower motor neuron makes its way out the ventral root to a skeletal muscle. Okay, so know where the cell bodies are. Upper motor neurons in the primary motor cortex, lower motor neurons in the ventral horn. Know where the pathway decussates. The corticospinal tract decussates at the decussation of the pyramids in the medulla oblongata. And know the cell body of the lower motor neuron is in the anterior horn. The axon of the lower motor neuron goes out the ventral root, out the spinal nerve, to a muscle. I think we are good, you guys. That was a little bit longer than I had intended. So, so get your lab quiz in sometime this week, please. You will have a group quiz just like the last one. You guys have already submitted your answers for probably on Monday for this material. I'm sorry, Wednesday for this material. So don't worry about that for a little while. I know there's kind of a lot of different things going on right now in the class. I just want to be clear, guys. My goal is to have a quiz for lab and lecture every week. We may not have one every week. I'm very casual about the quizzes, very casual, especially if I make a mistake by posting it late or something like that, or being confusing in an announcement. What I'm more serious about is the midterm and the final. So the purpose of the quizzes is just for you to get some practice. I hope that everybody gets hundreds on these. They're not worth very many points, so even if you don't, it's not a huge deal. But your grade is really truly based on the midterm and the final. You'll have one for both lecture and lab. So as we get a little bit closer, I'll start talking some more about that. We'll do a Zoom call to discuss and see if you guys, just want to talk any concepts over. So for now, just my advice is to watch these videos, practice on the app. When you take a quiz, treat that like a study opportunity. Have your book out, have your notes out, study, learn as you take the quizzes. And I think you guys will do great on the test. So I will talk to you guys next week when we cover, oh boy, what is next week? Arteries. We're going to be talking about arteries of the upper extremity. So see you then. Thanks.